Good morning, everyone. Thank you for joining us today. In our last discussion, we dove deep into how nanotechnology is really building the future of computing. Today, we're making a fascinating shift. We're moving from silicon chips to the very building blocks of life itself. We're going to be exploring the incredible intersection of nanotechnology and human health. And I'm thrilled to have an expert joining us to help unpack how all these ideas, which might sound like science fiction, are actually becoming today's clinical reality. So let's just dive right in. First up, we're going to lay the groundwork. What is the core principle of nanomedicine? What does it actually mean to engineer solutions at the scale of life? From there, we'll see how that thinking leads to these hyper-targeted treatment systems that are just revolutionizing drug delivery. Then we'll turn to diagnostics. I'm talking about sensors so sensitive they can spot a disease before you even feel a thing. We'll also touch on the mind-blowing world of regenerative medicine before we land on what is really the ultimate proof of concept for this entire field, the story behind the mRNA vaccines. Okay, let's get right into it with that first piece, establishing the core principle behind all of this. So, to start us off, let's get to the heart of it. When we say nanomedicine, what is the fundamental idea? What's the core principle that really drives this whole field forward?
Well, the core principle is actually beautifully simple in its concept, but it's incredibly powerful in practice. You have to think about it like this. Our bodies are these amazing machines, and they're governed by processes that happen at the cellular and molecular level. Diseases, when you get right down to it, are often just malfunctions in this nanoscale machinery. So the idea of nanomedicine is, well, if you want to fight a disease that starts at the molecular level, your tools have to be engineered at that same scale. For centuries, medicine has often used, let's say, a sledgehammer, when what it really needed was a scalpel. Nanotechnology finally gives us that scalpel, the ability to design and build solutions that can interact directly with the machinery of life. And what's so crucial to understand, as of today, is that this is no longer science fiction. It's moved firmly from a futuristic concept into clinical reality, and it's solving some of the biggest challenges we face in healthcare.
That's a perfect foundation. And that analogy, moving from a sledgehammer to a scalpel, that's really powerful. So let's build on that and jump into our second section, which is all about applying that precision to how we treat diseases. You mentioned that sledgehammer approach, and that really hits home when you think about certain treatments. So what is that biggest problem with traditional drugs, especially powerful ones like chemotherapy, that nanotech is so uniquely positioned to fix?
The central problem is, in a word, specificity, or rather, a lack of it. Take chemotherapy, for example. It's designed to kill any cell that's dividing rapidly. Now that's great for targeting tumors, but it has no way of telling the difference between a cancer cell and other healthy, fast-growing cells, like the ones in your hair follicles or the lining of your digestive tract. So the drug just floods the entire body, causing all this collateral damage. And that, of course, leads to the severe side effects we all know about. But it's also a serious clinical problem, because that very toxicity limits the dose a doctor can safely give a patient. And that means the treatment might never reach its full cancer-killing potential. Nanotechnology completely flips that script by turning drug delivery from a systemic flood into a precision strike.
That makes so much sense. You want to maximize the effect on the target, but minimize it absolutely everywhere else. So how do these nanoscale delivery vehicles actually pull that off? Could you walk us through how they work, maybe starting with one of the most established systems out there, liposomes?
Absolutely. The core idea is to build essentially a tiny biological armored car. This vehicle wraps around the drug, protecting its payload from being destroyed as it travels through the bloodstream. And, most importantly, it's engineered to deliver that payload directly to the target, like a tumor, and release it only when it gets there. We have a whole toolkit of these vehicles now, but three of the most important ones are liposomes, nanocapsules, and dendrimers. Each one has a pretty unique structure for different jobs, but they all share that same fundamental goal: targeted delivery.
So let's zoom in on liposomes, because they're a fantastic example, and they're already being used in FDA-approved drugs. A liposome is basically a tiny, hollow sphere made from a double layer of lipids. It's like an artificial cell membrane. And this structure makes it incredibly versatile. Its hollow, water-filled core is perfect for carrying drugs that dissolve in water, but at the same time, you can embed fat-soluble drugs right into the lipid membrane itself. They're also biocompatible, which means our bodies generally don't see them as a threat and attack them. And here's the final piece of the puzzle. We can actually stud the outside of the liposome with molecules, like antibodies, that act like a key. This key is designed to fit a specific lock found on cancer cells, which lets the liposome dock directly onto its target and then deliver its payload.
That's incredibly elegant. I mean, the ability to carry different types of drugs and actively target specific cells, that's a huge leap forward. So what about those other systems you mentioned, nanocapsules and dendrimers? How are they different?
They work on a very similar principle, but they use different materials and structures. Nanocapsules, for instance, are tiny polymer spheres, but they're often designed to be smart, engineered to break down and release their drug only when they sense a specific trigger, like the acidic environment you typically find around a tumor. Dendrimers, well, they're even more wild. They aren't spheres at all, but these precise, tree-like structures. The branches create all these little pockets to hold drug molecules, and the outer surface can be customized for targeting. They just offer an incredible degree of control over the final design.
So we've got these highly sophisticated delivery systems. But beyond just delivering traditional drugs more effectively, how is nanotech being used to directly attack cancer cells? Let's kick that off with an approach called photothermal therapy. This is where things get really fascinating, because here the nanoparticle itself becomes the weapon. The whole process is just ingenious. First, gold nanoparticles are injected into the body, and they're designed to accumulate inside the tumor. Then, a surgeon aims a near-infrared laser at that tumor. Now, this specific wavelength of light is harmless to us. It just passes right through healthy tissue without doing anything. But the gold nanoparticles are engineered to be perfect absorbers of this exact light. So when the laser hits them, they instantly convert that light energy into intense, localized heat. And they effectively cook the cancer cells from the inside out, all with minimal damage to the healthy tissue right next to them. It's a way of delivering targeted thermal energy on command. Wow, that's an incredible concept. Using light to remotely activate a destructive force exactly where you want it? It sounds like there's a similar principle at play with another technique I've heard about, magnetic hyperthermia. How does that one work?
It's a very similar idea, just with a different energy source. Instead of gold nanoparticles and light, this method uses superparamagnetic iron oxide nanoparticles, which are basically tiny specks of rust that have special magnetic properties. Just like before, they're guided to accumulate inside a tumor. Then an alternating magnetic field is applied from outside the body. This field is completely harmless to the patient, but it causes those iron oxide nanoparticles to flip their magnetic orientation back and forth thousands of times a second. All that rapid flipping generates intense heat right where they are, again, killing the tumor cells with incredible precision.
So we've got this amazing new arsenal for treating disease once we find it, but that really begs a critical question. How can we find it earlier in the first place? And that's the other side of the coin, which brings us perfectly to diagnostics. Right. We all know that catching things early is absolutely critical for successfully treating so many diseases. So how exactly is nanotechnology helping to create the kinds of sensors that can give us those crucial early warnings? The core challenge really is all about sensitivity. When a disease is just getting started, its biomarkers, those telltale proteins or bits of DNA, are present in such tiny, tiny amounts that conventional tests just can't see them. They just miss them. Nanomaterials, though, have these unique optical and electrical properties that let us build biosensors with truly unprecedented sensitivity. They can hunt down and flag these biomarkers even at incredibly low concentrations, which means we can see the invisible signs of a disease long before any symptoms appear. That ability to detect just trace amounts really feels like the whole game. Can you give us a concrete example of that? For instance, how are nanomaterials called quantum dots used as these biological labels? Quantum dots are a perfect illustration of this. They're these tiny semiconductor crystals, and their standout property is their fluorescence. They glow brilliantly when you shine UV light on them. What makes them so special for this job is that their glow is incredibly bright, and it's also incredibly stable. It doesn't fade quickly like traditional dyes do. You can even tune their color just by changing their size. So in diagnostics, you can take a quantum dot and attach it to an antibody that's been engineered to seek out one specific target, like a cancer biomarker. When you introduce this into a patient's tissue sample, the antibody finds its target, and that glowing quantum dot acts like a brilliant beacon, clearly lighting up the location of the disease, even when there are very few of those biomarker molecules present.
So quantum dots are like microscopic searchlights. That's a fantastic way to visualize it. Now, let's talk about a technology that a lot of us have actually used at home. How do gold nanoparticles play a role in creating those rapid diagnostic tests that we're also familiar with now? This is really a perfect example of nanotech in our daily lives. The reason those tests work is because of the intense ruby red color of gold nanoparticles. In a test for, say, COVID, these tiny red particles are coated with antibodies that are designed to grab onto the virus. When the virus is present in your sample, it gets snagged by these particles. As everything flows along the paper strip, the particles that have caught the virus get trapped on the test line. And suddenly, you have enough of these intensely colored particles all concentrated in one place that they become visible to the naked eye. And that's your positive line. It's a simple but brilliant piece of optical engineering.
It's really amazing that such a common device relies on such advanced principles. So we've talked about these optical labels. What about materials that are more known for their electrical properties, like silicon nanowires or carbon nanotubes? How are they being used in this space?
Those materials are the basis for what might be an even more sensitive class of electronic nanosensors. You can build a tiny circuit using a silicon nanowire or a carbon nanotube, and then coat its surface with molecules that will only bind to one specific biomarker. Now, when a single molecule of that biomarker from a sample latches onto the nanowire, its physical presence actually disrupts the flow of electrons through the wire. This causes a measurable change in the wire's electrical conductivity. And because the binding of even one molecule can create a signal you can detect, these sensors hold the promise of achieving what is essentially the holy grail of early diagnostics: single molecule detection.
Single molecule detection. That's just mind-boggling to think about. Okay, so we can detect and we can treat disease with this incredible new precision. But what about rebuilding what's been damaged by disease or injury? That takes us into another amazing frontier: regenerative medicine.
Exactly. I mean, the ultimate goal here is to repair or maybe even replace damaged tissues and organs. So, how is nanotechnology contributing to the field of tissue engineering to help literally rebuild the human body?
The fundamental challenge in tissue engineering is that you can't just put a bunch of cells in a petri dish and expect them to grow into a functional organ. They need a supportive structure, a scaffold, that tells them how to grow and organize themselves. Inside our bodies, this structure is called the extracellular matrix, or ECM, and it's this incredibly complex network of fibers with nanoscale dimensions. The nanotech solution is to create artificial scaffolds that precisely mimic this natural ECM. We can now create these mats of nanofibrous material that provide the perfect environment to guide cells to grow into functional tissue. This is already being used for things like engineered skin grafts, and for building scaffolds to help bone and cartilage repair itself. We can even take medical implants, like a hip replacement, and coat the surface with nanoscale textures. This creates a bioactive surface that encourages your own bone cells to grab on and integrate with it, which can lead to faster healing and a much more stable, longer-lasting implant.
That's incredible, using nanoscale blueprints to actually help rebuild the body. This brings us perfectly to our final section, which discusses what many people are calling the ultimate validation of all these principles we've been talking about: the nanomedicine revolution.
Right. You really can't have a discussion about modern nanomedicine without talking about the COVID-19 vaccines. They are called a nanotech triumph for a very good reason. So to understand why, what was that fundamental show-stopping challenge of using messenger RNA that nanotechnology was absolutely essential to solve?
The central challenge was the mRNA molecule itself. It's an absolutely beautiful piece of biological software. It carries the instructions our cells can use to produce a protein, in this case, a harmless piece of the virus that trains our immune system. It's incredibly powerful, but it is also incredibly fragile. I mean, the human body is just filled with enzymes that are ready to find and destroy any foreign RNA they encounter. If you were to just inject raw mRNA into someone's bloodstream, it would be obliterated in seconds. It would never even stand a chance of reaching a single cell. So for decades, mRNA was this brilliant idea with no practical way to make it work. It needed a delivery vehicle. So the software was written, but
but there was no way to actually run it on the hardware of the human body. How then did a very specific piece of nanotechnology, the lipid nanoparticle, or LNP, provide such an elegant and effective solution to that problem?
The lipid nanoparticle was the absolute breakthrough. The effective mRNA vaccines were just as much a triumph of nano-delivery technology as they were of biology. These LNPs perform three critical functions. First, and most obviously, they protect that fragile mRNA inside a protective lipid shell. Second, they stabilize it, allowing it to travel safely through the bloodstream. And finally, and this is the crucial last step, they are designed to fuse with our cell walls to deliver their mRNA payload directly inside. The global success of these vaccines was just a resounding, undeniable validation for the entire field of nanomedicine. It proved that this delivery platform was safe, it was effective, and it was scalable on a global level.
It truly was just a remarkable convergence of decades of research, all coming together at the perfect moment. So, taking everything we've discussed today, from that precision drug delivery and ultrasensitive diagnostics to regenerative medicine and these incredible nanovaccines, what is the overall impact? What's the significance of nanotechnology when it comes to revolutionizing medicine?
The overall impact is that nanotechnology is fundamentally changing the entire paradigm of medicine. It's giving us an unprecedented toolkit to interact with the very machinery of life itself. The successes that we've seen, especially recently, have moved this entire field from the realm of promise into life-saving reality. So it's not just about creating incrementally better treatments anymore. We are truly architecting an entirely new era of healthcare, one that is more predictive because we can detect disease so much earlier, more personalized because we can tailor these treatments to individuals, and ultimately more powerful than anything we've ever had before.
That is an incredible way to put it, and thank you so much for breaking all of this down for us. It's just amazing to think about the precision to attack a disease while reducing side effects, the sensitivity to detect it earlier than we ever thought possible, the ability to help the body regenerate itself using these nanoscale scaffolds, and, of course, that undeniable validation that came from the mRNA vaccines. It really does all point to this completely new era of personalized, predictive healthcare that isn't just coming, it's already here. The implications really are staggering. We hope you'll join us next time as we continue this series and we'll explore nanotechnology and energy and the environment.
Thank you again for joining us for this fascinating discussion. If you want to keep exploring how technology is shaping our world, be sure to subscribe to our channel, Tech, AI, and Beyond, so you don't miss our next one.